Nice to see so many familiar faces here. I'm changing a little bit gears here. I'm not part of the software development team. I'm part of the operations team, so basically the COO portion of Biognosys. And in this function, I'm heading the laboratory that we have just outside Boston here. And I'm very happy to present you some data on a collaboration that we had together with Johns Hopkins University. And it's basically a showcase also how large-scale proteomics of biofluids can be used to identify biomarkers. And of course, the software part plays also into that. Primarily because I forgot to actually add the disease to the title that we're working on. It is Alzheimer's disease, which probably most of you would have guessed by looking into amyloid beta converters. Just to bring everyone on the same page, it's a progressive brain disorder that gradually destroys memory, thinking skills, and at the end, the ability to perform daily tasks. It's a most common form of dementia with 60 to 80% of the cases. At least up to now, it's irreversible and degenerative, and it primarily impacts neurons, leading to the cell death and brain shrinkage. There are two hallmarks of it. One is beta amyloid plaques that are primarily extracellular, and tau tangles which are formed intercellular, and basically lead to a dysfunction of the neurons at the end. There are probably more of it. It's a far more complex disease than I now put on this slide, but I just wanted to mention these two proteins as they are also important for later on in the presentation. So why is the early detection of Alzheimer's disease so important? So therefore, you can see on this slide certain the time frame of certain biomarkers and so on that can be used in clinics, and also give you a little bit an idea that it was able to early on basically detect onset Alzheimer's. The most prominent one is clinical function, so basically some of the symptoms I mentioned already on the previous slide. But by now it's actually able to detect it in a very preclinical stage with measuring amyloid 42 accumulation in CSF. But this can already be seen as like the first onset of the disease. So basically the first biomarker that we see might be already the disease developing. And unfortunately, there's not really a time frame on the clinical disease states, but as you can imagine, there are already decades of pathology accumulating, even if you detect like early accumulation of amyloid beta 42 in CSF. And therefore, actually, it's very critical to prevent the disease eventually, and also to look into potentially healing it, to find early biomarker, ideally from biofluids that can be routinely screened for. So for this study, it was important basically to decide early on which of the patients in the cohort would develop the disease. Therefore, we use amyloid beta converters, and there to define those patients, there are two measures that have been used. So one is in CSF, the amyloid beta 142 to 140 ratio, and also the p-tau 181 status, which actually leads basically, or is part of forming of the tau tangles and neurofibrillar tangles in the early disease state. So the ultimate goal is here, or we hope to find, is basically to find biomarkers that basically helps us to stratify patients, to find like to find patient that show like p-amyloid conversion basically before what's commonly possible at the moment. And for that, we turn to analyze CSF, which is kind of the most obvious biofluid because it's directly readout of the brain, if you want to say so, and also in plasma proteomics because it's like the most available biofluids. For that, we collaborated with Johns Hopkins University and worked on the BioCard study, which is a longitudinal resource for early Alzheimer's discovery. Within the study, which started in the mid-90s, administered at NIH. In 2009, it was taken over by Johns Hopkins. And within this cohort, there were CSF and plasma samples collected of more than 350 participants over 30 years. And at regular time points, there were also like cognitive tests performed and so on. So basically, it's a very well-described cohort. And at the end, also, some of the patients developed Alzheimer's. So basically, then it's possible to basically go back to the earlier samples and to see where did something develop differently, for example, in the plasma or CSF. From the overall cohort, 110 participants were selected for this kind of proteomic study. And as you can see, there are several time points for each patient involved. So in total, we had like 433 plasma and 566 CSF samples. Within the analysis, we also included a couple of QC samples. Overall, we had like a sample set of more than 600 samples for both body fluids. As also probably most of you know, biomarker discovery in plasma especially is very challenging. So it's a very diverse pool of analytes. So for example, there can be proteins in it released from blood cells, cell leakage proteins, actively secreted proteins, and so on. And actually, it also has a very broad dynamic range. So in the highest abundant range, you see like typically like consider classical plasma proteins. Then in the lower abundant range, it's like tissue leakage proteins, which are potentially interesting as readouts of disease. But also interleukins, a very interesting class of proteins to look at. And overall, that covers like almost 10 orders of magnitude, which is actually not possible to be analyzed by current mass spectrometry approaches, which I can also further highlight here. So what you see in neat plasma, but it's also to a minor extent true for CSF, which has a little bit less high dynamic range, but still a dynamic range that exceeds what's accessible with the mass spectrometer. So you basically typically see the highest abundant proteins. Traditionally, what was done, basically using an antibody-based depletion of proteins, so basically removing the highest abundant proteins from the samples, and basically you can access like the middle abundance range of proteins. And within the last years and so on, it became more popular, use particle-based enrichment approaches. It's probably not 100% correct that you really deplete the highest abundant proteins, but at least you reduce the proportion of these proteins in the overall sample so that you can basically squeeze more proteins into the dynamic range that you can analyze with the mass spectrometer. Our plasma enrichment workflow is called P2. So in brief, it can be seen that around this nanoparticle there's a hard and soft corona formed, which basically leads to enrichment then of proteins and therefore also de-enrichment on the same hand of high abundant proteins in the plasma samples. If you look at some test samples that we did early on when we developed this material, compared to neat analysis, P2 basically allows you to identify or can quantify up to 10 times more proteins than with neat analysis. So with that approach, we're actually able to get a high coverage of plasma and CSF proteome, but there's still another challenge of analyzing hundreds of LC-MS runs, which was also already kind of addressed by some of the previous speakers. I put here one picture that most of you probably have also seen on the largest study we analyzed, actually was 3,000 samples, and that basically is symbolized here with the boxes on the EvoSep, which also then allows like a very reproducible IC separation, and also the timsTOF HT is a very robust instrument to analyze this data set. So in this study in particular, we had like 627 plasma files, which was overall 1.8 terabyte of data. The CSF file, we had a little bit fewer samples because we have had fewer control samples in the CSF sample set, but still it was also a sample set of 1.6 terabyte. And actually, then the solution to the analysis is, Tejas actually gives the proper introduction for that already, is using then OmicsFlow and the cloud-based analysis of it. So basically analyze it in parallels, not one file after the other. And the plasma set was analyzed in around 6 days. The CSF set was faster in around 3 days. And these were also early versions of it. Current versions would be a little bit faster than that, but it also depends a little bit how you design your cloud setup. So how many parallel processes you allow. So there's actually also the potential to be even faster than that. One other thing that I was quite curious about is also whether it would be also possible then to use it for PTM analysis. For that, we used like an extended set of PTMs that we would expect in plasma or CSF. And it was also possible to analyze this data set. It took like roughly double than the normal analysis, but at least it was possible, and actually also showed some interesting insights that at this point, just an overview of how many of these PTMs we identified in the different sample sets. We still have to look further into what might be the biological meaning of it, but it was actually quite nice to see that some of the modifications we saw, for example, oxidation of methionine is like expected, but it was also like interesting to see that the spread of these modifications seems to be a little bit higher in plasma than in CSF samples. But what that means, we still have to look further into it. But at least we are now able to actually look also in this layer of the information for larger data set. Coming back to the matched CSF plasma sample set. So we did the enrichment for the plasma using P2. For CSF enrichment, we used a different enrichment approach. But the basic principle behind it is the same. We used a 40 samples per day method for both sample sets. So we basically analyzed both sample sets in around 2 weeks on the mass spec. And for the data analysis, as mentioned, we used OmicsFlow. For this plasma data set on median, we identified around 7,000 proteins, and also 6,800 of the proteins were identified and quantified in at least 50% of the samples. But there were also some samples where we identified just a very low number of proteins. The CSF data set, we identified in total and on median less proteins than in the plasma data set, primarily related that the enrichment workflow is not as efficient as the enrichment workflow used for plasma. But on a median, it's still almost 3,900 proteins per sample, so still delivering a good coverage also of the CSF proteome. Before we actually wanted to go further into the biological analysis of the data, we also looked at some of the technical variants and how they might influence the data. For that, we used the variance partition analysis, here shown for the CSF samples. What you could see here is actually that the main influence of the variance is explained by the different patients across time, which is a good outcome because we want to follow like the biological differences within the patients. We see some effect of preparation plate, but that was a little bit related to our experimental design because we basically put the same patient on the same sample preparation plate to minimize any technical variance within these time series for a patient. What was very interesting to see actually is like what we call collection batch. So we basically saw no influence whether the samples were taken at NIH or at Johns Hopkins. This was actually very nice to see. That this will not influence the results too much. For plasma, it looks a little bit different. So the patient ID still has like the largest influence on the variance, but we saw a larger influence on the collection batch, so basically where the samples were taken. So that might be a little bit an indication that the standardization of plasma collection is a harder task than of CSF collection. And again, we saw the same effect from the preparation plate, but again, it's related to the experimental design, which was similar for the plasma and the CSF cohort. Then we were also quite interesting. So we are looking at samples that are 30 and more years old, whether we see any effect on the storage time for some commonly used like quality markers for plasma. So we looked at platelet contamination, erythrocyte, and PBMC contaminations. And we didn't see a large difference for NIH and Johns Hopkins samples. But if you look very closely in it, for example, you see a slightly higher erythrocyte contamination score for the samples that were collected at Johns Hopkins. But overall, it was not like the older the sample was, the higher these contamination scores. We also looked at the CSF samples. There we basically just checked whether the number of quantified proteins was the same depending on when the samples were collected. And there you could see that there's a slight drop maybe in the median identification if you look at samples collected in NIH and compared to Johns Hopkins. But again, there was no trend visible that the older the samples, basically the less proteins we identify. So that seems to be rather constant over time. And then also we looked again the storage effect more in like a principal component approach, where we basically try to understand whether we see like any like batch effect based on the age of the samples. But what you can see here is basically independent on when the samples were taken. Basically all of these distribution of points are basically on top of each other. So there was not storage time related batch effect on the global proteome visible. Then the first time then we applied a linear mixed model to basically find proteins that potentially allow us to define amyloid beta converters. So the first linear mixed model we applied was not considering the age of the patient. There we actually find a very high number of differentially abundant proteins across the sample cohort. The same was true for plasma and CSF. We found like 500 proteins that changed in the same directionality in plasma and CSF, but we also find 500 proteins that changed in the opposite directionality. But again, that might be just related that the CSF is a readout of the brain health, so to say, whereas the plasma of the whole body. So there might be other secreted proteins actually that lead to a different directionality of the fold change. And it has also been described in literature actually that the age of the patient has a large influence on the plasma and CSF proteome. Therefore, we find the linear mixed model, we adjusted it for age, and then we could actually narrow it down to some proteins that would basically help to predict amyloid converters and control groups. So some of the proteins are highlighted here. I also want to give you a little bit. Then we also did a functional annotation analysis to basically figure out whether they're related to some specific function. It was actually very nice to see that amyloid-related terms showed up among the top enriched pathways. So actually that indicated that the markers we're looking at are potentially relevant also for the disease. This slide is a little bit busy, but I want to highlight a few things. So what we basically wanted to look at here is like the proteins that show up as significantly changed between amyloid beta converter and to the control group. And basically in red you can see how this protein changed over time in the amyloid converter cohort, and in green in the control cohort. And what we're particularly interested in where these lines basically cross, because that would be basically like an easy way to basically define that is basically where you could start diagnosing the disease using these proteins based on the data we generated here. And in yellow are highlighted actually proteins where our lines cross before actually the clinical defined amyloid conversion age. So these are potential biomarkers that would allow to define these groups before it was possible clinically. To give you a little bit more like focused view on two examples here. So RSB, it was clinical conversion age was around 70, and if you basically look in our set, so we don't see any change in this protein over time in the control group. In the amyloid converter group, it starts accumulating, and if you again look at the crossed lines in plasma, it precedes around 8 years the conversion age. If you look in CSF, it's actually coming down from 70 to late 50s, so it's actually even earlier potentially possible using this protein to detect amyloid conversion. And the same is true for this antibody below. There we can see that actually in plasma, the median amyloid conversion age that was defined clinically is very close to what we see based on the proteomics data. But if you move to CSF, we can actually see that it is much earlier there as compared to the clinical readout. We also looked a little bit into some more classical markers for Alzheimer diagnosis. So tau is a very prominent protein here. There we can basically see, if you just looked at total protein tau, we don't see a difference in the levels basically in plasma, but we can see that actually the total levels were increasing in the amyloid converter cohort over time as compared to the control group. Yeah, with that I come to an end. So with the state-of-the-art plasma and CSF workflows, we are actually able to enable a deep proteomic analysis of hundreds of samples. As I mentioned, so the largest core sample we worked on was also in the range of 3,000 samples, so even 600, and can be moved further. OmicsFlow is very important for the analysis because these large sample set and analyzing them in sequential mode is almost impossible. And with unbiased proteomics in matched CSF and plasma proteomics, we can find molecular changes that precede the clinical amyloid conversion by years. We're still working on further confirming that, but actually the first data on that look very promising. And the last finding was here that CSF, the proteomics that we see in CSF, is far earlier than what we see, or for most protein, it's earlier than what we see in plasma, which is also not 100% surprising, as you can see that CSF is closer to the brain than the plasma, but it's also a very interesting finding here. With that, I would like to thank our collaborators in Johns Hopkins, especially Abe and Ida, and my team in Newton, Alan and Nicole, who worked on the sample set. Paulina and Johan were basically then also involved into the data analysis and also in setting up this collaboration. And thank you very much for your attention. Yeah.
Very nice, Jan. Thank you. What changes, if any, did you have to? What changes, if any, did you have to? What changes, if any, did you have to make in the P2 protocol in order to use it for CSF?
So we used a different particle. We tried it also with P2 particle, but the yields were too low. So we basically ended up with a few hundred, less than 100 nanogram per sample, and then we cannot get a high coverage. So we used a different particle type, which is not as efficient in enrichment, but has higher peptide yields at the end.
And you're making that particle commercially available?
No, we actually hope to develop further P2 that it works on CSF. That is kind of our way forward.
You also mentioned that phospho-tau at 217, which is in fact now a clinically deployed marker for Alzheimer's. You said you worked on PTMs. Did you see any phosphorylated peptides from tau in your analyses?
That is something I need to further look into it. I finished the analysis basically 2 weeks ago and didn't have the time to dig that deep into it, but that is exactly the question we are interested in, and in general, which phosphosite we see of tau, which also have like described any function.
Yeah. And then just a comment, I think to consider using CSF as a preclinical diagnostic for Alzheimer's is probably a non-starter. People are not going to get CSF draws. So in fact, even that test that I mentioned, they're not even thinking about deploying that until cognitive function is shown as being declining.
Yeah, I can totally see that. There's also, the point is to use CSF basically as a kind of model, discovery model, so to say, and hope that we can find similar changes in plasma. That is the idea behind.
Hi, just continuation of the question. So my understanding is that for the matching plasma and CSF, you didn't use P2. It was the modified where you got 9,000 or 5,000 proteins.
Correct. For plasma, we used a different particle, yes.
Again, both cases, it was not P2. It was a different...
For plasma, it was P2. But for CSF, it was a particle we call P1, which is kind of an earlier version, but has higher peptide yield, but less good enrichment.
Plasma, you got 9,000 proteins with P2.
Yes, in total across all 600 samples.
Okay. Thank
So Jan, very nice talk. So I have some technical questions quickly. So for the P2 enrichment, what is the volume of plasma samples you have to start from? And also related to that, for plasma-based biomarker studies, normally you need to consider either you normalize your data to the volume or to the total amount of the proteins that you load per injection. And if you normalize that to the volume, of course, it's more translational to the clinical samples. Can you share some experience in that regard?
So to the first question, we start with 100 microliter at the moment routinely. But the idea is also to optimize the protocol further to start with less. But typically for human plasma, 100 microliter is typically available. So that's not so limiting. It's only limiting if you want to move to mouse, for example. Traditionally, we actually normalize everything to the protein content. We are looking into it also further, especially also given that there are other proteomics methods now, like immunity-based methods that are basically based on volume normalization, or most of them at least. That's something we're still looking into it. I don't have a good answer at this point, so only that we use traditionally like total proteome normalization, which is also like the pre-assumption of the normalization that then applied in SpectroNaut. So we use like the standard normalization in SpectroNaut as well. But that is something we're discussing actually quite a lot at the moment, how to normalize this data best and whether it might be better actually to always inject like the same plasma volume instead like same protein amount. That's a very good question, yeah.
I had the same question about CSF. How much CSF are you starting with for these experiments?
That was 200 microliters, so rather high amount volume.
I have a quick question about the platelets contamination because we're always discussing if this interfering with the quantitation of your target. You have some of the patients seem to have a slightly higher platelet contamination than others. Did you see a trend where you seeing your target? So you think that contamination played a role, or you see it was independent?
So it's a good question. So we have to. So we haven't particularly looked into whether some of these changes are driven by platelet contaminations. That is something we also still have to further review, actually, the candidates we get out of it, whether it's like on one of the contamination panels, so to say. We hope a little bit that with the high number of samples that we have, that it normalizes it out a little bit, so that we basically lose it like with the statistical testing. But yeah, we haven't particularly looked into that.
Thank you.
Hi. So I got a question about the LC. So for high throughput, so you're running continuously for 600 samples. Over time, do you see any like changes in like pressure in column? Do you have to change the column, or do you see any like change in signal after the first one and the last one? And how do you fix that?
So this sample set was like acquired within roughly 2 weeks, and there we didn't really see like a decrease in signal over time. For larger sample sets, of course, that becomes more prominent, but then we would try to do like routinely like some minor cleaning on the instrument. So that becomes more and more a problem the larger the cohorts get, of course. And in this 200 sample, it's not like such a big problem yet. So you can basically run, or at least we could run them in one go and saw rather stable signals over time. Some of these samples sometimes can lead to clogging of a column. That happened not in this cohort, but in other plasma samples we run. And what we typically do then is basically, depending on how many samples run on one or the other sample, would reacquire samples. It was just clocked after 5 samples, would reacquire everything on the same column. Otherwise, we will also look then whether the column had a batch effect on the resulting data, and then would correct for this batch effect if it's like happens in the middle of the sample. But it can have an influence, that's certainly true.
Okay, so thank you very much, Jan, for your talk.
Thank you.